Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine a global health crisis, just relentless,
Mhm.
claiming over, 150,000 lives every year,
Yeah, it's staggering.
and so many children affected. We're talking about a disease that's getting smarter, resisting our best medicines.
Mhm.
Doctors are facing what one expert called the final warning sign, typhoid fever.
That's right.
But what if there's been a solution, maybe a simple one, right under our noses this whole time?
Well, that's exactly what we're diving into today. There's some really fascinating new research out of Cornell University,
Okay.
published in eBiomedicine, and it offers, well, a significant ray of hope, really, against this threat.
So what are we looking at? A new paper?
Yep, a scientific paper and a related health news article. We're going to unpack a totally new way an old antibiotic seems to work.
Okay, so our mission today,
Hmm.
figure out what this antibiotic is, how it works its magic in the surprising way, and, what it means.
Exactly. For global health, for maybe even you listening.
All right, let's start with the basics. Typhoid fever. Remind us.
Sure. It's caused by a bacterium, Salmonella Typhi.
Right.
And it's a life-threatening systemic disease, so it affects the whole body.
Not just a stomach bug.
No, definitely not. And it's a huge global health issue, especially South Asia, Southeast Asia, Sub-Saharan Africa. Still very much a present danger.
And the big challenge now with resistance, isn't it?
Oh, absolutely. It's becoming a massive problem. We're seeing this alarming spread of drug-resistant strains.
Like the MDR ones?
Yeah, multi-drug resistant, or MDR. Those resist the first-line antibiotics, ampicillin, chloramphenicol, cotrimoxazole, the classics.
Okay.
But then it got worse. We started seeing extensively drug-resistant strains, XDR.
XDR. So even more resistant.
Way more. They resist all the MDR drugs, plus fluoroquinolones and third-generation cephalosporins. These were our backup plan.
Wow. So what's left?
That's the scary part. In places like Pakistan, these XDR strains are widespread. Doctors are seeing cases where basically nothing works anymore.
That is the final warning sign.
It really is. It signals a critical point.
So what makes Salmonella Typhi so hard to fight? Beyond just resisting the drugs, does it have some kind of trick up its sleeve?
It does, yeah. A big part of it is its protective outer layer. Scientists call it the capsule.
A capsule. Like a little shield.
Exactly. Specifically, it's the vicapsular polysaccharide. Think of it like a very effective cloak. It hides the bacteria from our immune system.
Sneaky.
Very. It helps it infect us, helps it evade detection, and it can even block antibiotics from getting inside the bacteria properly.
So it's hiding and blocking treatment.
Pretty much. And to make things trickier, there are different variants of this capsule.
Help-rate.
These variants pop up in about maybe 1 in 5 cases globally. And there's one called hypervi.
Hypervi. Sounds bad.
It is. It's hypervirulent, much more aggressive, causes more severe disease, and sadly, higher death rates.
Okay, so we're dealing with a global killer, getting harder to treat, wearing this protective cloak. A really tough bug to beat.
Definitely formidable.
But here's the twist, right? This Cornell research, they found something.
They did. They identified an existing antibiotic, Rifampin, that turns out to be remarkably effective against Salmonella Typhi.
Rifampin, I've heard of that. Isn't it used for TB?
Exactly. Tuberculosis, traveler's diarrhea.
Yeah.
It's been around for ages. FDA-approved on the WHO Essential Medicines list.
And it works against typhoid. How well?
The study found it was, get this, 99.9% effective against Salmonella Typhi.
99.9%. Wow. That's
Yeah.
incredible. Why was it overlooked for typhoid then?
That's the surprising part. It was kind of sidelined, possibly because of concerns about resistance, especially with rifampin-resistant TB emerging.
Ah, okay.
But this new research checked clinical isolates, samples from actual patients, and found over 99.9% were still highly susceptible to rifampin.
So this effective tool was just sitting there, like finding out your old keychain as a hidden feature.
Something like that. It was hiding in plain sight.
So how does it do it? How does rifampin manage this against such a tricky bacterium? What's the secret?
Well, this is the really cool part, the core of the discovery. The researchers, Zhangmin Song and Bi-Young Lee, found it works through what they call decapsulation.
Decapsulation. So
Yeah.
it removes the capsule.
Literally, it strips off that protective outer layer we were just talking about, takes away the cloak.
It uncloaks the enemy.
Yeah.
Makes it vulnerable.
Exactly. Once that capsule is gone, the bacteria are suddenly exposed. They become susceptible to our own immune system.
Ah. so our body can then recognize it and fight it off.
Precisely. It makes it much easier for our immune cells to clear the infection. And what's really interesting
Yeah.
is that this happens even at lower concentrations of the antibiotic,
Yeah.
levels below what you'd normally need just to kill the bacteria directly.
So even a lower dose weakens them enough for our immune system to step in.
That seems to be the case. These lower levels, the kind you'd naturally get in a patient during treatment, are enough to trigger this decapsulation and boost our own defenses.
That's elegant, actually. It's like the drug is helping our immune system rather than just carpet bombing the bacteria.
Yeah, that's a good way to put it. A strategic partner.
Is this unique to Rifampin? Or do other antibiotics do this uncloaking thing too?
It seems pretty specific to Rifampin and its class, the rifamycins. They tested another common antibiotic, azithromycin, often used for drug-resistant typhoid, and it didn't show this effect.
Okay, so Rifampin's special. How does it work at the genetic level? How does it stop the capsule from being made?
Right, so it doesn't just strip off the old one, it stops new ones being built. It does this by inhibiting the expression of the genes needed to make the capsule.
Turning off the factory.
Basically, yes.
Yeah.
And other virulence factors too, the things that make the bacteria harmful.
But how does it target those specific genes? Is there something special about them?
There is. This is quite clever. The researchers found that the genes rifampin affects, like the ones for the capsule, and for something called Salmonella pathogenicity, island 1 virulence factors.
Okay, those sound important.
They are crucial for invading our cells. These genes have significantly higher amounts of adenine thymine in their DNA sequence.
Adenine thymine, AT, high AT content.
Exactly. This high AT content seems to make those specific genes more vulnerable to rifampin's action. It's like a genetic weak spot that rifampin exploits.
Fascinating.
Okay.
Let's shift gears a bit. We've got the lab stuff, the mechanism, but what does this mean practically for patients, for fighting these infections?
Right, the real-world implications. And the evidence is pretty strong. First off, rifampin worked against those nasty hyper-Vi variants, too.
The really aggressive ones.
Yeah, which is crucial because they're spreading globally. They showed this really clearly in mouse studies.
What happened with the mice?
Mice infected with that hyper-Vi variant, when treated with rifampin, had significantly lower death rates and much less bacteria in their organs, liver, spleen, gallbladder.
That sounds promising.
Yeah.
But how do we know it's definitely the decapsulation making the difference, not just rifampin killing bacteria?
Good question. They did a neat experiment. They took bacteria, treated them with rifampin just to remove the capsule before infecting the mice.
Ah, so the drug wasn't even in the mice.
Right. And even then, those mice had much higher survival rates and lower bacterial counts. It really showed the power of removing that capsule itself.
Okay, that's convincing. Did they look at human cells too?
They did. They used human immune cells, neutrophils, and sera. They found that the decapsulated Salmonella Typhi were much more easily engulfed by these immune cells.
Engulfed, like eaten up.
Yeah, a process called phagocytosis. And they also triggered a stronger chemical attack from the immune cells, a reactive oxygen species burst. Both are key ways our bodies fight off bacteria.
So it works in mice, and the mechanism seems relevant to how our immune systems work too. That's huge.
It really is.
And you mentioned earlier this might work for other bacteria too, not just typhoid.
Yes, that's another exciting angle. This mechanism seems broadly applicable. They looked at the genes for capsules and other important bacteria. Salmonella Typhi, Klebsiella pneumonia, Acinetobacter baumannii.
All serious pathogens.
Absolutely. And they found those capsule genes also have that high adenine thymine, that high AT content.
The potential Achilles heel.
Potentially, yes. And they actually showed rifampin could decapsulate Salmonella typhi as well. So this could be a new strategy against a whole range of nasty, encapsulated, drug-resistant bacteria.
Wow. It really broadens the scope.
It does. Co-author Malkin Song put it well. She said, let me find the quote here. Our study shows that rifampin would be one solution if there are very severe or difficult cases, such as hypervirulent Salmonella Typhi infections that are also multi-drug resistant.
Makes sense.
And she added, we hope that some doctors in the affected countries implement our finding to treat their patients.
A direct call to action almost. But, okay, as promising as this all sounds, science always has next steps, right? Limitations.
Yeah.
What needs to happen now?
Absolutely. It's never quite that simple. While rifampin is approved and available, its specific role, say, as a second-line treatment for typhoid, especially those fluoroquinolone-resistant cases, that needs more clinical validation.
So more human trials.
Likely, yes. And clear guidelines. We need established clinical microbiology guidelines for testing Salmonella Typhi's susceptibility to rifampin, like we have for TB.
Right, so doctors know exactly when and how to use it for typhoid.
Yeah.
Standard protocols.
Exactly. It needs to be properly integrated into practice safely and effectively.
Are the researchers planning more work? Or is this it for now?
Oh, they're definitely pushing forward. They mentioned plans to look at combinations.
Combinations.
Yeah.
Like Rifampin plus something else.
Yeah, combining decapsulation drugs like Rifampin with drugs that target the typhoid toxins, for example, trying to hit the bacteria from multiple angles.
Disarming them even more completely.
That's the idea, a multi-pronged attack.
This really does feel like a potential game changer. An old, affordable, available drug tackling a super-resistant killer just by figuring out this new way it works. It's kind of amazing.
It is. This deep dive really shows that sometimes the biggest breakthroughs aren't brand new molecules, but finding a new trick in an old dog, so to speak.
Yeah.
Understanding a new mechanism for a familiar tool. And it highlights how powerful our own immune system is if we can just help it see the enemy.
So a thought for everyone listening.
Yeah.
As you go about your day, maybe think about this. How many other solutions might be hiding in plain sight? Overlooked tools, resources, maybe even ideas in your own life, your work. They could do so much more if we just looked at them differently.
Yeah.
With fresh eyes.
It's a great question. That potential for rediscovery.
Definitely something to think about.
I agree.
Well, thank you for joining us on this deep dive into, well, a truly fascinating bit of science.
My pleasure. It's exciting stuff.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.